The topic is nucleocytoplasmic interaction. Let's see about the common thing between the nucleus and the cytoplasm of a cell. Nucleus is the most important part of the cell that is situated in the cell cytoplasm. All these cellular activities are controlled by it. Nucleus is directing and organizing unit without which the cell could not exist. The nucleus and the cytoplasm are interdependent and one cannot survive without the other. The cytoplasm provides the energy for the cell through the process like oxidative phosphorylation and anaerobic glycolysis. The cytoplasmic ribosome contains most of the machinery for the protein synthesis process. The nucleus provides the templates for the protein synthesis like mRNA, rRNA, and tRNA. Thus, there exists a balanced dynamic relationship between the nucleus and the cytoplasm during the process like cleavage, gastrulation, histogenesis, and cellular differentiation. At the level of interaction between the nuclear and the cytoplasmic compartments, the cytoplasmic state has been shown to affect the nuclear behavior and cell functions. And in different cytoplasmic or nuclear physiological state are implicated which are transmissible to their progeny. So this discussion of the nucleocytoplasmic interrelation therefore includes like the mechanism by which the genes contained in the chromosomes exert their control on the metabolic process of the cytoplasm, and the next is the mechanism by which the cytoplasm influence the gene activity. There are different types of experiments that is showing interdependence and interactions, that is control of nucleus over cytoplasm. In that first case we have protozoa, and as second case we have acetabularia, and as third case we have sea urchin. First let's see about protozoa. It was first noticed by Balbiani in merotomy experiments in which protozoa were enucleated and studied. So such enucleated fragments are able to sustain most cellular activities, and they can form a cellular membrane and carry on photosynthesis and react to the stimuli and ingest food, activate the cilia, etc. So these enucleated cells generally survive only for a short time, that is within 5 to 10 minutes, and they are incapable of growth and reproduction after 5 to 10 minutes. As observed in the amoeba proteus, that after the removal of cell nucleus, its about movement has been slowed down and it can no longer digest foods. When the nuclei are transferred from one amoeba to another of the same species, activation of the cytoplasm division and mass culture may occur. And when the nucleus of another species is implanted, the cytoplasm is activated, but cell division occurs less readily. So these experiments have shown that the cytoplasm of the cell cannot survive alone, and that activities are on same manner initiated and regulated by the nucleus. Next, in the case of acetabularia, J. Hammerling in 1934 demonstrated that the nucleus controls the function of the cells and ultimately the characters of the individuals by conducting experiments on acetabularia, that is a unicellular marine algae. That is, acetabularia has a stem between 3 to 5 cm long and a cap of 1 cm in diameter. The nucleus is located in the basal or rhizoid of the end of the cell. In acetabularia, the nucleus can be removed simply by cutting off rhizoid, and resulting enucleated cells are active in the photosynthesis phase and can survive for many weeks. Each acetabularia species have a particular cap morphology. That is, if a nucleus from the acetabularia crenulata is implanted into an enucleated acetabularia mediterranea, from which the cap has been removed, the resulting cell will be eventually developed as a cap of acetabularia crenulata, that is intermediate type. Further, if two nucleate cells of different species are grafted together, a hybrid having a cap of intermediate morphology is formed. This is the process that is taking place in the acetabularia, that is crenulata, intermediate, and mediterranea. So this result clearly demonstrates that the shape of the cap is determined by the nucleus. Although Dependent on the nucleus, cytoplasm has some degree of functional autonomy like storage of information in active form, which can later helps in the cap formation. Next we'll see about in the case of sea urchin. In sea urchin, an unfertilized sea urchin egg enucleated by being cut or centrifuged may be stimulated to divide without fertilization by slight immersion in the hypertonic solution. In the absence of the nucleus, it undergoes division to form a multicellular and haploid embryonic form, which later on regenerates, and once the nucleus forms into haploid and embryonic form. Apparently, division of this cytoplasm done in limited time and can continue without the presence of a nucleus, but a nucleus is necessary for a continued and normally functioning and differentiation of the cytoplasm of the cell. So, in conclusion, the above experiments have shown that the synthesis of the macromolecules in a nucleus is controlled by the cytoplasmic environment. That is, although nucleus plays an important role in a cell, the cytoplasm is the energy provider to the cell. Finally, both nucleus and cytoplasm are interdependent to each other, and one cannot survive without the each other. Thank you.